Our microbiology capstone group is called Oenologists Anonymous. We've partnered up with Dave Ashwanden of Chill Switch Winery from Cedar Ridge, Colorado. He makes natural wine with little to no additives. Dave uses Colorado-grown grapes near his vineyard in Cedar Ridge. In the fall, he crushes the grapes, removes the stems, and places them into an open vat to ferment with the natural microbes present on the grapes for 1 week. He then uses another batch of wine that has already been pitched with cultured yeast to inoculate newer batches and ferment for another month. This process is called backslopping. This year, the wine that he uses to inoculate others is Sauvignon Blanc, to which he has pitched an unknown strain of S. cerevisiae. When almost all the sugar has been converted to alcohol, he presses the grapes, removes the skins, and transfers his wine into oak barrels and then inoculates with wine containing a strain of S. cerevisiae called EC1118. This wine is left to age in the barrels for up to 30 months before being bottled. Dave adds a small amount of sulfite at this point, but otherwise no other chemical additives. For our project, we will test the following hypotheses. The first one, the microbial diversity of the wine will be observed to decrease throughout the wine production process as the less prevalent microbes are outcompeted by the inoculated species, primarily Saccharomyces cerevisiae. Our second hypothesis says yeast species, especially wild ones, will show reduced growth in the presence of antimicrobial, antioxidant, and pH-adjusting agents relative to lactic acid and acetic acid bacteria, such as those from the genre Lactobacillus, Acetobacter, and Gluconobacter. This selective effect will be more pronounced at lower concentrations of additives. To add to that, the antimicrobial and antioxidant agents potassium metabisulfate and potassium sorbate will reduce growth of all organisms more than the pH-adjusting agents, citric and tartaric acid. To test these hypotheses, we took samples of the wine at several time points throughout the process. We sampled 2 types of grapes and wine, Petit Verdot, a red wine, and Gewürztraminer, a white wine. We took samples from grapes straight out of the vineyard and must that had been fermenting with only natural yeast for several days, as well as must that had been fermenting for several days after the inoculation with another wine. Due to the timeline of our project, we are unable to get samples of the exact same wine throughout the process. However, we are able to collect samples of wine from 2017 and 16 that had been aging in oak barrels for 1 to 2 years respectively, as well as bottled wine from 2015 to which small amounts of potassium metabisulfate have been added. With each of these samples, we plan to extract DNA and have it sequenced to determine the microbial communities present. Some of the experiments we've done so far is an isolation of organisms on selective medias. We've T-streaked these to select for colonies, we've gram-stained, and we've spot tested different additives. The DNA extraction and sequencing will be our next step. We cultured organisms from Petit Verdot and Gewürztraminer musts that fermented with wild yeast. This was before backslopping inoculation, as well as from Syrah must that had been inoculated via backslopping. We isolated individual colonies and gram-stained the isolates to identify them as yeast or gram-negative or gram-positive bacteria. The organisms isolated from the Petit Verdot sample were exposed to the additives at concentrations ranging from 10% down to 0.01%, as well as 100% pure thyme oil used as a spot test. The isolates were streaked into a lawn and 2 microliter spots were pipetted onto the plates. After incubation, we measured sensitivity by looking at the zones of inhibition. Several of our results so far have been surprising. We did not plan on culturing organisms from inoculated musts, but cultured the Syrah simply to test out our methods. From these cultures, we observed greater diversity in the inoculated Syrah than in the uninoculated Petit Verdot. or the Gewürztraminer. This is the opposite of what we hypothesized, suggesting that the inoculation might actually introduce more diverse, viable organisms rather than outcompeting the diverse wild microbes. For the spot test, while all of our isolates were extensively inhibited by thyme oil, many were not inhibited by the other additives at any concentration. Only half of the isolates tested were inhibited by the highest concentration, 10%, of potassium metabisulfate, and only 3 of the 15 isolates showed partial inhibition by the 10% potassium sorbate. Although these results are surprising, they must be taken with a grain of salt given that the conditions on an agar plate are not necessarily representative of the environment in wine. As for the future of our project, we plan to extract and sequence both fungal and bacterial DNA from each of our wine samples to determine the microbial communities present at each time point. These results may support the use of thyme oil in winemaking process, and further research may test the effects of other plant extracts on the wine microbiome, as well as their effects on the aspects of wine quality such as taste. We were unable to test diluted concentrations of thyme oil since it is not water soluble, and while undiluted thyme oil was very effective in inhibiting growth, it is rather pungent and likely would not contribute favorably to wine flavor.